without further ado, I'll hand it over to Joanne.
Thank you. Thanks, Nicole. Thanks, Nicole and Matt for organizing the seminar series. I could get a lot out of it, and I'm hoping to get a lot of input from the work we've been doing on the microbiome in the past 5 years. So the idea of phenotypic diversification, not a new one. Charles Darwin wrote about it elegantly in On the Origin of Species, saying, "From so simple a beginning, endless forms most beautiful and most wonderful have been and are being evolved." And the simple statement has just been an obsession for evolutionary biologists in general, trying to understand what are the drivers, what is the basis that creates this wondrous diversification that we see all around us. So we know a number of basic things, a number of primary drivers. There's of course the habitat, the abiotic conditions of each habitat, whether it's temperature, humidity, elevation, all of these features will drive adaptation, will drive features of adaptation, such as having more fat, having feathers, having physiology that's adaptable to water or very cold or very dry climate and temperatures. There's also diet. So this can cause a lot of diversification, even within a single generation. One nice example of this is carotenoids that are taken up by various birds. So finch, Baltimore oriole, and this is a yellow shafted flicker. And the colors in their beaks and their feathers come from carotenoids in their diet. And this color can change if they happen to come across a different type of berry that's more appealing to them, or something that people like to plant a lot that's not endemic to the region, can also change their feathers from more yellow to orangish. And then recently, in the past 20 years, microbes, the environmental microbes, have been a significant factor in what can drive evolution and phenotypic change within a single generation. So but we haven't really incorporated microbes into our understanding of adaptation and evolution. So this has been one major aspect of the research that I'm going to talk to you about, is the role of environmental microbes on phenotypic diversification. So to do this, we used Hawaiian Drosophila, and these flies are actually a marvel of phenotypic diversity. The Hawaiian species account for 20% of the world's diversity in Drosophila, which is amazing. And here you see that they also inhabit various habitats with ranges in elevation from 600 to 1500 meters, steep gradients in rainfall, temperature, and evapotranspiration. These are just some of the sites where you can find these Hawaiian flies. And these are just pictures of flies that we've collected from each of these sites. And you can see that they have pretty spectacular differences in their cuticular patterns and pigmentation on the cuticle, as well as their wing patterns. And this is just the stuff we see by eye. There are also a lot of other really interesting aspects of their ecology and diversification. So for this reason, they make a marvelous model for trying to understand how microbes in the environment contribute to this diversification. I'll tell you a little bit more about diversity. Here again, you see some nice pictures showing the diversification of their wing patterns and cuticular patterns. They also vary in terms of what kind of host plants that they use for their diet, and also oviposition. So the larvae will eat these host plants, and the females will select only certain parts of these plants for laying their eggs. Some flies, some of the Drosophila are specialists, so they will only select one or two plants from each of these genera, and others are more generalists and will select from a variety of these plants. And just on a side note, each of these plants are endemic to Hawaii, and they also have some interesting cultural properties. And so, for example, Tushardia, the cord of it is really strong and is used for rope and to make fiber. Oha, the sap from this plant is used for is like, it's kind of like aloe, so it's kind of like this soothing gel. Wickstromia I find fascinating because the bark is actually contains a toxin. So it's used to stupefy fish when used for fishing. But somehow certain species of plants or flies are able to subsist on this Wickstromia despite this toxin. Another source of enormous phenotypic diversification in the flies is how they smell. So these are mass spectral profiles of their pheromone gland. Each of these profiles represents a different species, and each of the peaks in these mass spectra are different peaks, different lipids and small metabolites that are found in their pheromone gland. And you can see a lot of biochemical diversification from species to species. So trying to understand how all of this comes about, and to what extent the microbes can play a role has been one of the main aims of the work I'm gonna tell you about. The last feature of their phenotypic variation that's kind of fun is also their mating behavior. So there is Drosophila mccormickii, and you can see the male here. Doing this feeding thing initially, so that's called nuptial courtship, when it's nuptial feeding, when it's providing some kind of chemical cue to the female, and if she accepts, then she will allow him to copulate. She's not accepting, so he's gonna try again. You know, she's too busy eating, you know, so he's gonna try again. And he does this abdomen curling when this happens. But this kind of behavior that's very complex to set up because it involves song and audible signal, chemical signals, and visual signals. It looks really different for another species, Adiastola. So you see that they also do this nuptial feeding, but both the male and the female are doing this. And the male is also flicking his wings at the same time. So he's either fanning something forward or he might be singing a song. And then you watch his abdomen. So this one, the abdomen curls up and it actually exudes a droplet that the female will also imbibe, and it's the foreplay to their eventual copulation. So in one case, abdomen curling under, another case, abdomen curling up. And that's just the stuff we can see, knows what is going on chemically and in terms of their courtship song. So yeah, a lot of remarkable variation in these flies and their ecology and their behavior. Okay. So to address the question of how microbes contribute to this, we use two approaches. One is a more macro view of things. So it's to first characterize the microbiome of natural Drosophila populations and to see how much variation there is and what drives the assembly of their microbiomes in the wild. And second is to look at the functional role of the microbiome in their host fitness using manipulations in the lab. So we know from earlier projects that looked at a wide variety of host systems. This includes the Earth Microbiome Project on C-MΑIKI transect study and other studies looking at animals. That at least if you look at this really macro view, a number of factors have been identified as primary drivers of microbiome contribution, microbiome assembly. pH, temperature, salinity, what biome they're in, whether it's a host associated versus a free living community. From our transect study, habitat type and trophic levels, all of these are really important factors in being able to predict how the microbiome assembles. In a survey of 24 animal species, this is from Seth Bordenstein's group, they found that microbiome profiles fell closely to phylogeny, but this is looking at pretty diverse types of animals, so by host clade. Genetics plays a role in who gets to be in your microbiome, as well as the environmental features that are available to you. So which of these categories will Drosophila microbiomes fall into? So we looked at phylogeny first. So on the left here is phylogeny of the 35 Drosophila species generated by Carl Magnaca and Don Price. And we looked at how the order of the flies according to their phylogeny matched with the order of the flies according to their microbiome profiles. So this is ordered by phylogeny, and then on the right here is ordered according to how similar their microbiomes are. And you can see a lot of crossing over of the lines, indicating that it's not really following phylogeny. So I'll just point out an example here. So Drosophila solifera and Drosophila aureus, they're pretty closely related according to phylogeny. But you can see that they switch positions on this dendrogram of the microbiota when you look at it this way. So from this, what we can conclude is that there is a lack of phylogenetic clustering, and there's limited evidence for co-evolution between the fly species and microbes. So just because they're more genetically related to each other doesn't mean that they're going to have a more similar microbiome to each other. So if it's not phylogeny, then what's driving their microbiome assembly? So this is from a study of 35 endemic species, over 500 samples from major islands in Hawaii. And so from alpha diversity, and only the fungal profiles were different between the islands. So this is the alpha diversity measurement, Shannon index. And the groups here are ordered according to the islands on which they were collected. So going from the oldest island to the youngest island, from Hawaii to Big Island. And you see no difference amongst the bacteria, the 60-ness profiles from island to island. So it doesn't seem to have any correlation with island age. In terms of the fungal profile, there is a difference between the oldest island, Kauai, and Big Island, and the youngest, the Big Island. But then there's this weird dip in the middle from Oahu. So it's not clear what's contributing to that. I mean, one obvious difference is the increased urbanization of Oahu. So island age, not really a major driving factor for their diversity. What about other factors? What about other abiotic factors? So this analysis was done by Sean Scoville from University of Wisconsin at Madison. And he did a multivariable matrix analysis of matrix. And what he found is that elevation, evapotranspiration, and island age were the strongest drivers for bacteria. And then for fungi, elevation, rainfall, evapotranspiration, and interestingly, phylogenetic relatedness. So for the most part, it looks like abiotic factors are driving the assembly of these microbiome profiles. Even though their diversity is similar from island to island, the actual composition of these profiles is very different between each of the different islands. Yeah, so we wanted to take a closer look at this overall trend. So rather than looking at this 535 species, we wanted to focus on a couple of species for which we had a good sample size number and which we could find in locate sites. So this allows us to do the following comparison, whether the same species but in a different environment has a more similar or different microbiome profile, and whether different species but placed in the same environment will have more or less the same or different microbiome profile. And what we found is that same species, so genetics are the same, but different environment leads to different microbiome profiles. Different species, but within the same local environment, similar microbiome profile. And then this is a comparison of beta diversity. So I'll just point out, so we're looking at Sproti and Tinitrix at Tom's Trail, Ka'u, and Ola'a. So Sproti and Tinitrix, two different species, both collected at Tom's Trail, see no significant difference in both its bacterial or its fungal profiles. If you look at the same species but collected in two different sites, so Sproti and Tom's Trail, Sproti and Ka'u, you see different profiles in terms of their beta diversity. So that's for this set. We see the same pattern also comparing Sproti and Tenitrix that were collected at Ka'u. Same microbiome profiles, but if you compare Sproti, same species from two different sites, you see a different microbiome profile. So this is again strong evidence that it's really the local environment that is shaping the microbiome assembly of these flies. So this is what we've established based on the study. So the local environment shapes the fly microbiome, maybe by altering the microbes that are available to them on their host plants and diet. Flies pick this up. But the big question is whether this actually leads to the phenotypic diversification. So this is the big question that we try to answer by looking at the functional role of microbiome using lab manipulations in Hawaiian Drosophila. So our method to manipulate the microbiome is to first to see whether microbiome plays a role at all. So we added either antifungal, antibacterial drugs, or both antibacterial and antifungal drugs to their food or control, which had nothing. And then we placed Drosophila grimshawi under these different dietary conditions. And then we measured their life history and traits. So we're placing them in as adults. So what we find, so to show you some of the major findings from this, a lot of this work was done by Matt Darris and Don Price's lab. So flies, we looked at the fecundity of females, so we counted the number of mature eggs in females. So under control conditions, you get usually around 80 to 100 eggs per female. But when you wipe out their microbes, you get almost no eggs are developing in these females. You could say that, okay, maybe it's the drug that's doing something off target to the fly. So we tried to rescue this fecundity defect by adding back a microbiome transplant. So these are active microbes that were collected from the frass of control flies, and we gave them back to the flies. Or we also inactivated the microbes by heating them briefly, and this would cause the microbes to cease making metabolites, and the flies could just be using them for food or nutrition. And what we find is that we are able to rescue somewhat by giving them back active microbes. Their microbiome profile also changes with these transplants. So this is the control fly. When we wipe out the microbes, we plate them, we don't see anything growing on MRS or YPB plates. But still they have a bit of microbes, and the proportion of them are primarily Rhabdomyosarcoma. And then when we rescue them, we get this rescue of gluconobacter back in their microbiome. And when we give them inactive frass, so that's microbes that are not active, we don't get this same reversal of the gluconobacter. So it might be that gluconobacter is pretty important to them being able to produce eggs and their overall fecundity. So this indicates that their microbiome is important for their fertility and fecundity. What about their courtship behaviors? So to record their courtship behaviors, we put a male and a female in this medium-sized petri dish, and then we put them under cameras, and then we set up all 4 conditions at once, looking at the ones where the controls, the rescues, the ones that have no microbes, the ones that don't have just been fed antibacterials, or the ones that have just been fed antifungals. So you got to do all of this in parallel. And we record them for 48 hours because they don't mate very much. And when they do mate, it only takes a minute or 2. And rather than making movies, because the files would have been enormous, we took a picture once a minute for 48 hours, and then had undergrads go through the pictures to look for population. So normally under these circumstances, when they have those normal microbiome in the lab, you get about 70% of them reporting here. For the most part, they just court once and that's it. They copulate once and that's it. When we wipe out the bacteria and they just have fungi, you also don't see much of a difference in their courtship and their copulation behavior. So the flies are still mating even though they don't have bacteria. When you do the complement where you wipe out the fungi but you leave the bacteria intact, then you see quite a decrease in female population. And when both pairs in this courtship chamber have undergone this treatment, then you see almost no copulation. What's interesting is that the males don't really change that much in terms of how often they copulate, but they do show increased copulation. So they have multiple occurrences in how many times they copulate. And that's indicative of maybe sensory signaling, something in their pheromones or something in the post reproductive changes that occur in the female to prevent her from rejecting a male after she's courted. So that's an interesting and kind of a unexpected result. And then finally, when we put in this chamber flies that have their bacteria and fungi wiped out, and you can see like a pretty big decrement in their overall population. Although again, the males are not that affected, and they are trying to copulate multiple times. So their behavior is changing, fecundity has changed. What about their lipids? So lipids are one of the major storage sites for energy, but it also shapes their reproduction, survivorship, lifespan. There are three main organs that shape the lipid profile. There's the gut, the fat body, which is this layer of cells that surrounds many of the organs, and the enocytes that are on the surface, the cuticular surface, they're just below the cuticular surface of the fly. And these enocytes produce waxes that make that shiny, waxy coating you see on insects. The function of these waxes are to provide desiccation resistance, protection from infection, and they can also function as pheromones and enable kin and sex recognition. So we first measured the cuticular lipid profile, and I'll just call your attention to what happens when we feed the flies antifungals. So actually, the amounts of lipids on their cuticle actually increases in both the females and the males. And why this is important because these lipids are related to desiccation. So you can imagine that if they have a thicker layer or longer lipids, waxes on their surface, they'll be better able to survive under warmer temperatures or drier conditions. So having this different cuticular lipid profile might actually increase their stress resistance to different kinds of ecological habitats and allow them to move into a new place. So that's the outside surface. What about the total fatty acid profile? So that's everything in the whole fly. So again, we see with the antifungal treatment that both males and females have an increase in overall fat throughout the fly. Total fatty acids, this is important measure for stress resilience, and it's also an important feature of reproduction. So any of these features that are important for the survivorship of the fly and moving on and reproducing could be affected by the microbiome, which appears to be a kind of rheostat that is altering these fundamental physiological features for the fly. So finally, I'll just show you these are cryosections that have been stained with phenol red, which is a general lipid dye. And you can see that in the fungal suppressed Grimshaw female, you can see a lot of these lipid droplets. Just to orient you, we're looking at the dorsal surface here of the fly towards the butt end here, and each of these round spheres here represents a lipid droplet, which store triacylglycerides, which are major energy molecule for the insects. So it's quite a bit thicker for the females. For the control females, you don't see much of that layer. So both the distribution of the fats as well as the amount of it has changed by altering their microbiome profile. And just to point out, these are the eggs of the fly here, and the fungal suppressed females don't have eggs. Yeah, so that's female physiological changes. We looked at some other changes in the male reproductive organs. So for males, they have testes that create sperm. They have accessory glands that produce proteins that accompany the sperm and are transferred to the females, and this helps to maintain sperm viability within the female. Some peptides are also transferred to the female that alter her behavior, making it more amenable for male sperm to be in there. So like their immune system gets changed with the transfer of peptides. And these peptides and proteins are produced in the accessory glands. Another important feature of the male reproductive organs is the ejaculatory bulb that produces lipids and components of the seminal fluid that also gets transferred to the female. And pheromones are also produced in the EB. So what we find for the male organs is that when we feed them antifungal drugs. The accessory glands reduce in total circumference. And they also reduce in area as well as the amount of protein that's inside of these glands. So these important proteins necessary for male fertility, those have decreased with the absence of fungi in their microbiome. We did a proteomics analysis to see exactly which proteins had gone missing. We found 145 proteins were downregulated after feeding them this antifungal drug, 28 proteins upregulated. A lot of these proteins have antioxidant properties, so they have to do with keeping the sperm healthy and viable when it's transferred to the female. So big changes for the male in terms of its reproductive capabilities. We also looked at the ejaculatory bulb. So this is the thing that produces lipid pheromones. And we see changes also. These are mass spectra produced on a product analysis in real time instrument. And you can see that when we feed them antifungal drugs, a couple of the major components drop away. This is just a volcano plot showing differences. Mostly we're interested in a couple of these. One is predicted to be a steroid. The other is a amino acid that might be used in their communication. So their pheromones are changing, their reproductive capabilities are changing, and their actual organ size and shape is also changing by manipulating their microbiome. But a lot of this we are doing by feeding them antibacteria and antifungal drugs. So basically hitting things with the hammer. And to what extent are there off-target effects? So really the cleanest way to do these experiments is to make azenic flies. And this is challenging with Drosophila because of the long lifespan and the long developmental time, as well as they don't lay that many eggs all at once. Melanogaster, you collect about 200 eggs, you bleach them, and then you let them develop inside the sterile conditions. With the flies, it was a challenge, but with the help of Iris Ridialou and Melisandre Pete, who was a postdoc earlier in the lab, we managed to come up with a protocol to do so. So this is azenic larvae, and you can see that they're much smaller compared to the wild type. But what's interesting is that they don't do very well without their microbes. So this graph here shows the survivorship and development of azenic eggs. We set up 20 vials. Each of these vials had about 50 eggs. For the most part, very few of them hatched. So we had a 4% hatching rate. And of the ones that hatched, that's the blue bars, only there's less than 1% viability. So the yellow bars here, which is basically one larvae for 4 vials out of 1,000 eggs survived. So this indicates that they desperately need their microbes to develop, even from the larval stage. This is different from melanogaster, where they're basically okay. They're a little bit slower, but they're actually okay without their microbes. But this is great because it gives us this blank slate which we can experiment, and we can start adding things back to the azenic larvae and see what's going to make them develop and grow up, and maybe even cause some phenotypic diversification. So what should we start adding back? We go back to their ecology. You remember that the flies lay oviposits onto rotting plants, rotting wood. And here you can see the eggs here that is inside the peeled away cambium of the rotting plant. And then this is a larvae that has emerged. So it emerges in this slimy biofilm of yeast and other fungi and whatever else is going on here. And this is the larvae's first exposure to the world is finding and eating all of these microbes that surround it. So in collaboration with Nicole Hinson's lab, Michael Fernandez, and Zane, in an undergrad in the lab, we've been collecting microbes, specifically yeast, from these rotting plants and culturing them to make a culture library. So this is just a small percentage of the yeast that have been cultivated and are currently being characterized. Kelly Konachek and Mike Fernandez, it's heroic effort, went out in the fields and collected plants at different stages of rot. So we found 5 native plant species that are known to be host plants for the flies. Made about 1,300 isolates and are so far trying to winnow it down and characterize this yeast, and you can just see a great deal of diversity. But with this library, then we can maybe engineer phenotypic diversity by using these different combinations of these microbes. Probably we'll have to add some bacteria in there, but it'll be really interesting just to see what is the impact of manipulating the yeast on its own, as well as the metabolites that's coming from them. Yeah, another way to try and narrow down which of these microbes might be important for their development and lifestyle is doing network analysis. So this is with a lot of help from Kirsten Nakayama and Laura Tipton. Laura was at UH, was a postdoc in Nicole Hinson's lab, and is now at James Madison University. So they created this network based on those 535 samples of native Hawaiian Drosophila. So each of the circles here, the nodes, represents a different taxa, and each of the lines connecting them, the edges, represent either a positive predicted interaction in blue or in red, negative predicted interactions. And we've thrown in here both bacteria as well as fungi. And the fungi are the ones down here, so the ones in the dark blue, and this purple, and then there is one fungi that has no edges, which is indicative that it's probably a tourist and doesn't belong here. So this is all correlative, but at least gives us a way to narrow down which microbes to focus in on when we are doing our manipulations. So I'll just point out a couple that are interesting. And also the Saccharomycetes and Tremolomycetes, a lot of those came up in the culturing. So we have a good library of these particular class of fungi. So if you look at these guys, number 65 looks like it has positive connections to bacteria, as well as to other fungi. So it has mostly blue connections. Whereas you look at number 70, so this is another yeast species belonging to Saccharomycetes. It has mostly negative interactions, but it also has some positive interactions with another member of the gamma proteobacteria. So this gives us a start to start finding our way into pairs of combinations of bacteria and fungi that might be important to helping the larvae develop and improving and altering the phenotypes of the adult flies. Yeah, so just to conclude, what we found from a survey of Hawaiian Drosophila across the Hawaiian archipelago is that environmental factors cause significant variation in host-associated microbiome. So it's not so much the genetics, it's the local environment. And then second, microbiome influences fecundity, mating behavior, accessory gland development, and lipid chemical diversity. So all the important stuff you need to grow up and survive. So currently, this is our thinking is that the local environment, microbes in the local environment get alter the fly microbiome assembly, and then based on the combinations of microbes in the fly, this will alter phenotypic diversity, survival, and reproduction. And this might just be one way how we get this endless forms most beautiful as it applies to Hawaiian Drosophila. Yeah, so just to conclude, just to thank people who did this work, mostly highlighted in yellow. Matt Medeiros, Vegas Matt Medeiros, I'm confused with Oahu Matt. Kelly Konachek do a lot of the Hawaiian Drosophila collection and rearing. Kirsten Nakayama and Laura Tipton helped the network analysis. Sean Scoville did a lot of the analysis for the Hawaiian Drosophila microbiome diversity. Ivis Ribrialo and Alex Samori are fantastic at rearing difficult species of flies and just kind of being patient and willing to try these difficult technical tasks of rearing like Asiatic Hawaiian flies. Nicole Hinson, Michael Fernandez will help with isolating geese and keeping them growing and happy and healthy. Yeah, and I think that's it. Yeah, thank you.